for coming, and thanks for that awesome introduction, Zach. Let's see if this works. So as
Hey, while he's getting the lights, it always
Oh, yeah.
presumptuous. I'm gonna have a party. I bought the food and the drinks. I'm assuming it's gonna go well. 7 o'clock. My house, which is 7 September Drive. Everybody's.
Okay, I think we're all good to go. So as physiologists in the Chevron lab, we study how animals use energy, and the ultimate goal for all organisms is to use that energy to maximize their fitness. Some very obvious ways they can do that is by reproduction or growth, but they also have to use it for things like maintenance, like this molting cardinal once a year has to replace all of his plumage to maintain, sort of integrity of the feathers, and that's a very energetically expensive process. And then also just a lot of times you're spending energy just to maintain homeostasis, right? Whether it's a panting dog or shivering in the cold, you're spending energy just to maintain your body temperature and keep your systems running. So today I'm going to be talking a lot about body composition, which is just the percent of fat and lean mass of an individual. And this sort of provides a snapshot in time of an individual's energetic states. So we can think of the fat mass or the fat component as sort of the fuel for the organism, and the lean mass is the engine. So the lean mass is muscle, organ systems, etc. And body composition is often estimated rather than measured. So you're probably familiar with for humans, the BMI, the body mass index. If you're familiar with it, you also know there's issues with it, and I'll touch on those a little bit later, but that's probably the most commonly known sort of measure of body composition or estimate of body composition. And one of the reasons why this trait is so interesting is to study is because how rapidly it can change. And it's these seasonal changes in body composition have been pretty well characterized in a few systems. Migratory birds are a big one, like this eared grebe that migrates from the northern plains, stages on the Great Lakes, and then moves down to the Gulf of California. So before they migrate, they actually shrink their flight muscles so much that they become flightless on the lake, and then they add 50% of their mass and fat before growing those flight muscles back, shrinking their leg muscles to take off and fly across the interior West. And this is happening over a matter of a couple weeks in the fall. Another well-known example is hibernating mammals like bears, and dormice can also add 50% or more of their mass in their weight in fat before entering hibernation. However, one thing we don't know as much about is how body composition varies across a species range, sort of in space more than in time. So there's not that much work on this. In humans, humans tend to be heavier and have higher BMI in colder environments. And then this example from starlings, they tend to have higher fat deposits at middle latitudes. And there's really not much work besides these few examinations of variation of body composition on a geographic scale. But because body composition is so critical to thermoregulation and a number of other processes, we have good reason to suspect that body composition should vary a lot, maybe a lot, across species ranges, especially if there's variation in temperature across that range. So I'm going to step through a couple of those reasons here. The first is that fats fuel a lot of aerobic activity. So in this case, shivering in particular is usually powered by fats, at least in some animals. And also subspecies such as mice and young humans have a special kind of tissue called brown fat that produces heat directly through non-shivering thermogenesis. So you're not burning it. That piece of tissue is actually just making heat on its own. Some species also use fat as an insulator to decrease thermal conductance and sort of lower their total thermogenic costs. And even outside of these cold environments, fats are a buffer for hard times. So if there's inclement weather or low resource availability, then you can sort of dip into those fat reserves to get by until the situation improves, or you can just get out of there. But carrying these intrinsic energy reserves around with you obviously comes with some costs. Most obvious one is just the extra cost of moving that mass around, like this bear that's about to go into hibernation is not moving too fast, probably. Another major factor is predation. So a lot of this is indirect. So generally higher predation pressure leads to lower fat reserves because of sometimes just stress responses or reduced foraging time in the presence of predators. But there can be direct impacts of body composition on predation. For example, birds with higher fat loads have slower takeoff times, which potentially makes them more vulnerable to predation. And then in small mammals, larger individuals in the population do not have access to as many refugia from predators as the smaller individuals in that population. And so today I'm going to be talking about body composition in the deer mouse system. So this is their range. They don't live all through this. This is not all their habitat, but this is what the CDC called it. So they are a great system for studying a lot of sort of physiological variation because they are all throughout North America in every habitat you can imagine. They don't hibernate, so they're active throughout the winter. The highest thermogenic costs. They're really easy to catch most of the time, sometimes. And they also take really well to captivity. So for a while, we had a big colony of mice here that were very happy living in the health science basement. So before I get into my data, I just want to pull back to body composition here. This is an important trait, but as I've alluded to, there's some major gaps in our knowledge about this trait. So I think a useful way to think about it for this talk is Stephen Arnold's framework of morphology, performance, and fitness roughly matches my 3 chapters. So my first chapter, I quantified how this trait varies geographically in deer mice. I next used an acclimation experiment to isolate environmental stressors and see how changes in body composition track changes in performance. And finally, I explored the complex relationship between body composition and survival. So my first chapter, how does body composition vary spatially? So we don't know a lot about that. We do know that how body mass varies geographically, right? Most of you probably heard of Bergman's rule, which is just the pattern that populations of animals in colder environments tend to have higher body masses or just larger body size. So this can be elevation or latitude. It applies to, obviously this is biology, so it doesn't apply to everything, but it applies to a lot of endotherms and even some ectotherms. And we don't really know how body composition contributes to this rule. So when an animal is adding mass, what sort of mass are they adding? So there's a couple different scenarios here. So I have mass splits into fat and lean mass here. So one option is to just increase the engine size, so higher lean mass in colder environments. I'm going to be obviously talking about elevation in this case, but this could equally apply to latitudinal gradients. Another option is just to increase your fuel tank and just increase your fat reserves. Or you could pursue a combination of these 2 strategies and increase both in proportion. And to get at this question, we conducted a big survey of deer mice across Colorado. So this is 27 sites. The dots represent the number of individuals caught there with the sex ratio. So we had about a 3,000 meter gradient over 27 sites with at least 10 individuals per site. So ended up with a little over 300 individuals total. And this was really fun field work. It took two and a half summers. But it's just to give you some more flavor of the habitat covered. There's obviously a lot of change over this gradient. So, we started in the warm shortgrass prairie in eastern Colorado, moved up into the foothills, then in the pine forest and all the way up above tree line at a couple places. So we collected on Pikes Peak and Mount Evans. So the highest places you can get to in Colorado by car. And of course, the fauna changes too, right? So it was really fun to go from, catching kangaroo rats and walking around prairie dog colonies down low to catching pika and having goats trying to break into the cabin up high. But actually, despite this like major turnover in animal communities, deer mice are one of the most common small mammals across this whole gradient. So in particular, at the high elevation sites and foothill sites, they're usually the most abundant small mammal, and they tend to be common in all habitats there. So that made things usually easier for running into deer mice. And so now I'm briefly just going to touch on how we measure body composition. This is the sort of how body composition used to be measured in the past, and one of the reasons why we don't have a lot of sort of modern measures of it. And this is a bomb calorimeter, where you basically ignite your sample and see how much energy that creates, and that's how you calculate body composition. So this is obviously low throughput, and you destroys your sample, so you cannot do longitudinal sampling, which is something I did not do in this chapter, but I'll do later. But fortunately, we have this newer method of QMR. It's similar to a regular MRI machine, except it gives you a ratio of fat to lean mass rather than an image. And it does that by taking advantage of the different amounts of water in fat versus lean tissue. So it can estimate about how much of each is in an animal. And so that the mouse goes in the tube here. This is a canola oil standard. So with standards, we're able to get within a tenth of a gram. One caveat is that it can't distinguish sort of within tissue type. So it can't distinguish between brown fat and white fat, or say muscle and organ tissue. So for most of my work here, after QMRing animals, I would dissect them and weigh the organs separately. And this, whenever I'm talking about measuring body composition throughout the talk, this is the technique I used. Okay, so the data. So deer mice did follow Bergman's rule as mass increased with elevation. As you can see, this is pretty noisy, low R squared, but this is pretty typical of, from what I can tell, of most sort of examinations of Bergman's rule. The females weighed more than the males and across elevations, and that was driven a lot by female reproductive activity. So I lumped females that were in any reproductive state together for the purposes of this plot. So either pregnant or lactating, they tended to be a lot larger. And digging into body composition, we see the reason for that difference in mass between the sexes is that the females have higher fat reserves. And again, this difference is maintained across elevations. And they add about, on average, a gram or a gram and a half of fat from the low site up to the highest site. However, lean mass is the same between the sexes. So that this also increased with elevation, but there is no difference between the males and females here. So that difference in overall mass is totally driven by fat mass. So given that, we wanted to see how body composition actually changes, and it really doesn't, because both lean mass and fat mass are increasing at about the same rate. So if we look at just percent fat, so fat mass divided by body mass, we see that really the only factor that's driving that is sex. So females have a higher percent fat than males, but that doesn't really seem to vary with elevation. So this supports the sort of balance prediction of fat and lean mass both increasing in roughly equal proportion. However, if we dig deeper sort of into that fat compartment, we see changes underneath the hood too, where brown fat, both sort of as a total mass and as a relative, as a percentage of their total fat stores, increased with elevation. So again, this is the special fat tissue that produces heat directly rather than being burned for energy. And there's no difference between the sexes here, but we did see a pretty substantial change in the amount of brown fat across the survey. So although here we found fat and lean mass increase in equal proportion. This is just one study and we need more to determine whether or not this pattern is sort of generalizable or applies more broadly. It could be that the deer mice are unique, or it could be that they have it's a matter of balancing costs versus cost of lean mass versus having enough storage on hand to run that the lean mass. Also, the higher relative and total brown fat at high elevation likely points to increased non-shivering thermogenic performance in those populations. So that brings me to my next chapter, where I look to see how do animals adjust their body composition to meet environmental changes, and not just body composition, but measure a number of performance-related traits here. And before I go on, I just want to briefly touch on sort of high altitude environments. So obviously you have both cold and the amount of oxygen available to you changing as you go up high. I'm going to use hypoxia as a term to describe sort of a lack of oxygen or a high altitude habitat. But these two pressures may have opposing effects. So cold acclimation usually way increases metabolic demand because you're increasing lean mass, muscle mass to maintain your body temperature in the cold. Whereas hypoxia acclimation actually suppresses appetite and leads to weight loss. And this is a pretty common phenomenon in mammals. Even mountaineers going up to base camp at Mount Everest, when they're just sitting around, will be losing weight because they have reduced appetite. And obviously with my first chapter, these two variables change in concert. So there's no way to sort of disentangle which is acting on the mice here. So we use a acclimation experiment. First, I just want to touch on the traits we measured. So maximum metabolic rates this is just what it sounds like. You put a mouse in a cold challenge and see how much energy it can generate. And we know that this is under selection in at least some high altitude populations. We also measured hypothermia resistance. So how long can individuals defend that body temperature? And that's especially important because you have to keep your body temperature stable to be able to function, right? So if your body temperature dips below, if you get hypothermic, you're basically toast if you're a wild mouse. So we're interested in how these traits change with acclimation and how body composition contributes to these. And we also were interested to see if populations responded to these treatments differently. So we had populations in the lab from top of a 14er in Colorado, Mount Evans, and then also from Lincoln, Nebraska. And after a couple generations in the lab, these maintained a couple of physiological differences. So they did not differ in body composition, but the Mount Evans mice were larger, a couple grams larger than the lowland mice, similar to the pattern I found in chapter 1. They also had higher maximum metabolic rates in captivity. So there seemed to be some both environmental and genetic differences between these populations. And we used a paired acclimation experiment. So these are hyperbaric chambers. The tubes go to a vacuum pump in the other room. We put plexiglass on either side and suck the air out to simulate high altitude. And they are inside an environmental chamber, so we can play with the temperature as well. So I had 4 treatments. I think there was a little turnaround, but warm normoxia is essentially the control. So that's room temperature in open to the environment. So Missoula elevation. We also did warm hypoxia with the simulated altitude, but still at room temperature. And then we did both those treatments at 5 Celsius. And we used almost 100 animals, F2s, 10 to 15 per treatment. And we used only males just to minimize variation in body composition going into the experiments. And they were individually housed, so they couldn't like huddle together as you often see, wild animals do in the cold. They were on their own here. And took a lot of measurements for this. I'm not going to talk about most of these, but weekly I measured fat and lean mass in the QMR along with food consumption before and after the acclimation. measured resting metabolic rate and the maximum metabolic rates. And then at the end of the acclimation, I measured body temperature, that body temperature challenge, and then also dissected the animals out to look at organ masses. Okay, so first results for maximum metabolic rate. This is the change in trait value from week 1 to week 6. So I said that the lowland mice or the highland mice have a higher maximum metabolic rate, but that's not what we're looking at here. We're just seeing how much each population changed across the acclimation. And as you can see, there was no population effect here. Both the highlanders and lowlanders increased their maximum metabolic rate in response to cold and hypoxia, with a particularly strong response to the combined treatment. There was no interaction there, but yeah. They also added lean mass under cold, but there was this interesting pattern where they did not add as much lean mass under cold hypoxia compared to cold normoxia. So we have this negative interaction between hypoxia and cold, again in both populations, where they added less lean mass than their normoxic cold treatment. And I'm not going to present the data here, but this was also mirrored in food consumption. So the cold hypoxic animals ate less food than the cold normoxic animals. And so this is kind of interesting because the cold hypoxic animals in the previous graph still had the highest or the largest change in maximum metabolic rate, even though they added less lean mass. And we think that this is probably driven again by brown fat. So here we had a positive interaction between cold and hypoxia. Again, no effective population. Both of them added more brown fat in the cold hypoxic treatment than the cold alone. And these are not changes in brown fat because you can only measure that once in an animal. Next, we wanted to look at this sort of high-level performance trait. How does the ability to stave off hypothermia vary? So again, because the highland mice have a higher maximum metabolic rate overall than blue, they're able to maintain their body temperature for longer. So this is minutes in the cold challenge, and then the probability of staying at their starting body temperature. So when it drops below 90% of starting body temperature, it goes to 0. They're hypothermic. At that point, we'd end the trial. So we had a population effect here. But the interesting thing was that the populations also responded differently to treatments in this trait. So unlike other traits, we see lowland mice only responding to cold. So these are both cold. Sorry, these labels are a little small, but this is cold hypoxic and cold normoxic over here. No difference between the warm hypoxic and warm normoxic treatments. Whereas the highland mice responded to both cold and hypoxia and responded particularly strongly to hypoxia. So this is the cold hypoxic and this is warm hypoxic here. So the highland mice responded really strongly to that hypoxia cue, even more so than the cold. So to wrap this section up, no, these populations didn't seem to change their body composition differently. They both increased brown fat in the cold and hypoxia, sort of similar to what we saw in the wild. Or we did not recapitulate other patterns of body composition from the wild, right? So acclimation increased lean mass. I didn't show you this data, but fat mass actually decreased over the acclimation rather than both increasing like we saw in chapter 1. Could be a number of reasons behind that. They had free access to food. It was also obviously a shorter time scale than the wild mice. And high hypoxia acclimation increased highlanders' ability to defend body temperature, but not lowlanders. And this has been shown in a few other traits before, but not this one, where the hypoxia or the highlands population can respond to hypoxia, but the lowlanders cannot or don't. Okay, so far I've shown you how deer mice change their mass or their body composition across their range and how that affects performance. So now I'm going to move on to the final chapter, which is how does body composition affect survival? And of course, survival is not the only component of fitness. We know that female body composition is a major driver of reproductive success in a broad array of taxa. So it influences, so increasing female fat reserves are associated with heavier offspring, with higher offspring survival, with larger clutch sizes, or even the decision whether or not to breed at all that season. I won't be talking about that much today. It's really hard to measure reproductive success in wild mice. So I'm focusing on the sort of other half of this fitness coin here, where you have to survive to be able to reproduce, right? So we want to see if body composition affects survival. And in addition to this, the straight survival aspects, a lot of studies have used condition indices. Remember this, that's like the the BMI, the estimate of body composition. A lot of studies use those to estimate sort of individual health, and basically saying if you have a higher condition score, that means you're in better health, and even expanding that, saying, well, this population has a higher score than the other, so this habitat might be better over here. So we're interested in looking at both how body composition affects survival and looking at effects of habitat. We do know something about how body composition affects survival, or condition affects survival. As you might expect, there's a number of studies that found higher condition, higher survival, particularly in the winter. Like this is this paper about white-tailed deer in the northern winters, that animals with greater fat reserves had a greater chance of survival through the winter. There's also some situations where there's apparently some sort of threshold where if you fall underneath that threshold, there's basically no coming back from it. So in this study, there was a El Nino year, I think, on the Galapagos, and the algae basically didn't bloom or didn't grow that year. So there was a major die-off of marine iguanas, and basically all the ones that died were underneath a certain condition score. And then in some situations, there seems that there might be some sort of stabilizing selection on body size. So in this case of wintering female mallards, animals in average condition had higher survival than those in low or high condition. So for my study of survival, I worked on the MPG ranch just outside Missoula for 2 years. I just want to stop here briefly to thank MPG Ranch for allowing access to this. It's a really awesome place. So thanks, Phil Ramsey, for that. And I want to thank Dean Pearson for helping get us on there and getting funding for it, and also sharing some of his data on the ranch. It's a really cool area. They're doing so it's no longer a working cattle ranch. They're doing these big restoration projects, trying to get rid of non-native species. So I had 2 plots. The first one is the 2 upper left, your left, upper left photos. So this sort of dry grassland, sort of sagebrush grassland, really nice in spring, of course, with the balsam roots. And this was my other site, and this was a restored site. So they hadn't developed anything on my primary site, but this one, a couple years before I came in, they did a prescribed burn and seeded it with a bunch of native plants, and then they provide supplemental water over the summer. So it's always green and much higher, much sort of denser vegetation than my other sites. And each site was 1 hectare with traps placed every 10 meters in a square. So 100 traps per site. And we trapped 4 nights a month, generally May through October, since you can't access the sites under snow for 2 years. After we captured mice, we would drive them back to Fort Missoula, to the flight lab, measure them in the QMR there. We'd give them a unique ear punch so we could identify them later, and then release them back into the wild. So we had only measured body composition once per trapping period, but obviously we were catching individuals many nights in a row, right? But we had about 250 total individuals and 400 different sort of QMR points, and averaging about 20 animals per capture period. Okay, first I'm going to talk about condition. And both condition and body mass were pretty weak predictors of percent fats. So this is something that people have actually found in lots of systems. That's people often use condition indices without validating them to see if, like, how much variation in these fat reserves they're actually explaining, and they often don't explain a lot, which is the case here. So I'm just throwing this up here to say be cautious with using condition and interpreting condition, and there's been increasing call to sort of move away from condition to more direct measures of body composition like I have here. The other advantage of that is that it allows for direct comparison between studies and taxa. So condition indices are normally mass divided by some index of size, either leg length or height or foot length. So that means it's really hard to say what condition in, white-tailed deer means compared to condition in deer mice. Where here we measure percent fat, we can say, okay, they have X percent fat versus Y. To assess survival, we used a couple different models. The main one was a Bayesian survival model that incorporated detectability. And we tested condition and mass, but also tested lean mass, fat mass, percent fat. And then I also broke fat into quantiles. So, I mentioned with the mallard example, how you might have higher survival for average fat scores. So this is how we got around that by treating fat as sort of a factor variable. And I don't have a fancy graph for this, but none of these affected survival here. So this was only a 2-year study, and detecting effects of selection in wild populations is not always easy. It could be the case that selection is only acting on body composition at certain times of year or during really harsh conditions. So to get at that, we also looked at a subset of data that was just over winter survival. For this, I had about 80 mice captured in fall sampling periods. And we just compared the mice. So we compared the measurements of mice in the fall. Sorry, I need to back up. So we captured them in fall, measured them, came back in the spring and saw if they were still there. And here we didn't include any measure of sort of detectability. We basically just assumed if we did not recapture you, you had left or died. But we did not find any difference in body composition between the recapture and non-recapture sort of populations here. So in fall lean mass, there was no difference between recaptures, spring and recaptures and non-recaptures, and same in percent fat. And finally, I'm just going to touch a little bit more on habitat. So again, we had this restored site with supplemental water and the undisturbed site with no water added. And we did find higher lean mass and fat mass at this site. So this is broken down by sex, because much like in my first chapter, the female deer mice in MDG had higher fat reserve than males. And then we have the restored site in blue and the undisturbed in red. And in both sexes, mostly the restored site had individuals were in better condition. So they had both more fats and, or rather higher percent fats and more lean mass too. Okay, so I'm gonna wrap up here, just sort of what did we learn from this? In terms of morphology, we found that both fat and lean mass increased with elevation, so no change in gross body composition, but an increase sort of in the subcompartment of fat with increasing brown fat. Similarly, in an acclimation experiment, we see that brown fat increased under cold hypoxia, and that could maybe be compensating for this blunted increase in lean mass we see under cold hypoxia. And then finally, body composition did not affect survival, but there was variation between habitats of different quality. And now just do some quick acknowledgements before I take questions. Of course, I need to thank Zach for being a great